Hello everyone, welcome to GPAT Can. So this is the recorded lecture class for your crash course and the regular batch students also, where we are going to cover a very quick revision on the analytical pharmacognosy. As I say, analytical pharmacognosy is very important for your exam point of view, for your competitive exams. So we are going to cover the analytical pharmacognosy with the mnemonics and in the digester part. So firstly, the drug evaluation is the main part of the analytical pharmacognosy, and you all know that the morphological or organoleptic evaluation is there, the microscopic evaluation is there, physical, chemical and biological evaluation are the part of the analytical pharmacognosy. So morphological or organoleptic evaluation, you know that for the special structures of drug, the color, odor, taste, shape, size, etc. are the part of the morphological evaluation. Mainly the organoleptic evaluation consists of what? Organoleptic evaluation is the evaluation of drug that is done or that is pursued by any kind of organ related feature. Organ related feature means what we can feel with our organs, like the color, odor, we can feel with our sensory organs. So that is the morphological or organoleptic evaluation. Next comes the most important part, that is the microscopic evaluation. And the microscopic evaluation is the leaf constant or the diagnostic character of leaves. That comes the first. Then comes the stomata, the calcium oxalate crystals, and the trichomes, etc. These are the main part of your microscopic evaluation. And the leaf constant, what are the leaf constants? The characteristic of the leaves, like the palisade ratio, that is average number of palisade cells beneath each epidermal cell. So epidermal cell is there, and beneath each epidermal cell the number of palisade shell is the palisade ratio. Vein islet number, there is a midrib and the margin. I told in class there is a midrib and the margin in the leaf of the so if that is a leaf, then this is the midrib and this is the margin. So in between this portion, the number of vein islets per square millimeter. Let us assume this is one portion. This is one square millimeter portion. So here the number of vein islets and the vein termination numbers are the vein islet number and the vein termination number, like how much veins are terminating there. Okay, next is the stomata. Stomata number is the per square millimeter, the number of stomata present in the epidermis of leaf. Next comes the stomatal index. It is very important, and I told repeatedly that this must be covered in your practicals also, in the semester exam practicals. So percentage which the number of stomata formed to the number of epidermal cells. So formula is the stomatal index equals to number of stomata divided by number of epidermal cell plus number of stomata into 100. This is the stomatal index. Next is the stomata. So the stomata, moss type, gymnospermous type, gramineous type, dicotyledonous type and circulus type stomata. So we have all covered it with the pictures and the examples and what are that. So most important is the dicotyledonous type, and we have to cover it in whole. And for the rest of the characteristics for your moss type, gymnospermous type, check your previous slide that I have given already. And for these things you just cover your definition and the examples. So the dicotyledonous type stomata is divided into these types. So these are the dicotyledonous type of stomata. And parasitic, see from the pictures that what are going to be there. So in the parasitic or the rubiaceous type, there are guard cells and subsidiary cells are parallel to each other. In the diacytic, they are in right angle to each other, the guard cell and the subsidiary cell. For the cruciferous type or the anisocytic, iso means same and iso means not similar. So guard cells are 2, but the subsidiary cells are 3, and one subsidiary cell is smaller than the other 2. So this is the aniso thing coming. One is smaller than the other 2. So not similar. 3 are not equal in hubs. Next is the anomocytic or the ranunculus type. Here the stomata is surrounded by varying number of subsidiary cells, and in the actinocytic or the radiating cell stomata, there are 2 guard cells are covered by circulating or radiating subsidiary cells. So these are the classification and definition or the characteristic you can say for the type of stomata, type of dicotyledonous stomata. And examples, for example, I have told the tricks. For the example, Senna and Coca and Ruby are parallel to each other. So Senna and Coca are the example for the rubiaceous stomata, and they are parallel. Guard cells are parallel to the subsidiary cells. Ments and tos is thyme. Vasaka gives cooling action. Cooling is the caryophyllaceous or diacytic. Vinca's belly dance is so cool and good. Vinca belly is belladonna, dance is datura, so is stramonium, and cool is cruciferous. Cruciferous is the anisocytic stomata. For the boycott, for the anomocytic, that is the boycott digital phone Oppo and Lamborghini and Redmi. So boy is buchu, cot is clove, digital is phone, ph not, f for phone. So F is Fenon, Oppo is opium, Lamborghini is lobelia, and Redmi is ranunculus stomata. For the actinocytic, Berinepia beer and starts acting. So this is the beer berry and actinocytic stomata. So for the trichomes, next, this is the classification for the trichomes, and there are the covering and non-glandular covering or the non-glandular trichomes and the hydratodes and the glandular trichomes also. So for the covering and non-glandular trichomes, these are the covering on the non-glandular trichomes, and I told that lignified trichomes is very important, and you can see the previous year question also containing the lignified trichomes example. So that is the Nux vomica and Strophanthus, and these are very important for your exam point of view. So covering trichomes or the non-glandular trichomes are divided into unicellular, multicellular unbranched trichomes and the multicellular branched trichomes. So basically they are divided into two, the unicellular and multicellular. Multicellular is further divided into branched and the unbranched trichome. So branched trichome is further divided into stellate, peltate, candelabra and T-shaped trichomes. So these are the example for the stellate, that is the hammer vellus, and for the peltate there is plate-like arrangement in humulus. Candelabra is for the verbascum thapsus, and T-shaped trichome is for the artemisia and the pyrethrum. And for the unicellular, there are lignified trichomes for the Nux vomica and Strophanthus. This is very important. Short, sharply pointed, curved cannabis, large conical lobelia. Short conical unicellular is for T and buchu, and strongly waved three-coiled trichome is for yerba and santa. And for the uniseriate trichome, there are bicellular, 3 cell long, 3 to 4 cell long, and 4 to 5 cell long. This was DSDB, Datura stramonium digitalis and belladonna, DSDB. So these are the uniseriate portion. For the biseriate, there is Calendula officinalis, and for the multicellular, there is male fern. And from this slide, the lignified trichome, is the most important. So don't forget that. Next is the glandular trichomes. Glandular trichomes are the unicellular glandular and the multicellular glandular trichome. If you want, then you can remember, or these are very complicated also, and I have not seen any question coming from there. So is there, we will cover that, but if you can, then you can cover that. But unicellular glandular trichome is for Piper, beetle and Vasica. And for the multicellular glandular trichome, there is unicellular head, unicellular stalk, unicellular head, uniseriate multicellular stalk. These are the examples. Mostly digitalis. Digitalis contains. So there was a question from the digitalis that which one is containing all type of trichome, like the glandular trichome and the non-glandular trichome also. So this will be the digitalis. Digitalis is containing 3 to 4 cell long non-glandular trichomes, and the unicellular head, unicellular stalk, glandular trichome, unicellular head, uniseriate multicellular stalk, glandular trichome, and the unicellular stalk, biseriate head, glandular trichome, and the okay, these three are only. So digitalis is the only plant that is containing both the non-glandular and the glandular trichomes. So next is the hydatodes. These are the organs of absorption or secretion of water developed in certain plants. So example is Piper beetle. So this is up to in this video, and in the next video we will cover the next portion for your revision of analytical pharmacognosy. So thank you everyone.